Once again, good morning everyone. As said in the morning, it's in the history of Science Congress, Kerala Science Congress, a Nobel laureate is giving a lecture in person, and I thank Professor Morten Meldal for agreeing to give this talk and then come here. Well, the responsibility given to me is introduce a Nobel laureate. I'm a little confused. What is an introduction required for a Nobel laureate? So I thought I'll just quote what the Nobel Foundation has scripted on their website about the finding of Professor Meldal. It says like this: chemists strive to build increasingly complicated molecules. For a long time, this has been very time consuming and expensive. Click chemistry means that molecular building blocks snap together quickly and efficiently. In 2002, Morten Meldal and Barry Sharpless independently of each other developed an elegant and efficient chemical reaction. The copper catalyzed acid alkaline cycloaddition. This is now in widespread use and is utilized in the development of pharmaceuticals for mapping DNA and creating new materials. And this is the invention for which Professor Morten Meldal got his Nobel Prize. I don't think he needs any other introduction than this. So thanks to you, and then over to you, Professor Meldal.
Thank you very much, and also thank you for the opportunity to talk not only about the chemistry that led to the Nobel Prize, but also a little bit about, you know, everything that is important for science and research. Because when you get a Nobel Prize, and I shared my Nobel Prize with Barry Sharpless and Caroline Bertozzi, Caroline Bertozzi, who I know since 2002 actually, but we have never worked together. Barry developed the click reaction for making small molecules react with each other. We developed it for larger macrocyclic macroscopic molecules, and Caroline Bertozzi developed the click reaction to be used in cells for very selective reactions with cell surface molecules, etc. When you get a Nobel Prize, you also get the right to come out with some statements. My statements are here. They are 4, and they are very important to me. The first one is everything is chemistry. Not all, but everything is chemistry. Everything is chemistry, meaning that we are chemistry. We are composed of molecules that interact. When we feel hungry, it's a hormone that signals to the brain that now we have to eat. When we feel tired, that's a molecule again. When we feel pain, it's bradykinin. When we feel very good, it's probably dopamine. So there's a lot of small molecular interaction in us that controls our psyche, our thinking, and our sensory system. But also all the materials around us, the sofas that you are sitting on here in the front, is actually made by human beings who have created molecules that goes together and makes this possible. So chemistry really is everything. And if chemistry is everything, it ought to be, in my opinion, part of the curriculum in school. Just like we are learning to read and write, we should learn about everything, and that is chemistry. I'll come back to how I think that is actually feasible in a global collaboration. Then, if we want really nice results in research, it's very important that the ideas is not the idea of a politician, but is the idea of a researcher himself. So therefore, the sandbox mentality of children, where they're really creative and making things in the sandbox, should continue into the grown research realm, where the researchers actually are looking for important things to follow up on, and actually can decide that now it's time to change track. I don't have to follow this track for 5 years because it's not going to lead anywhere, but over here there's a really good track that I should be able to follow. So therefore, funding by performance is the fourth of my mantras. I think those 4 mantras together could bring about a very efficient research environment with happy and satisfied researchers who would do the best they can for making progress on their research, not because they want a Nobel Prize, but because they are very interested in the chemistry that they are looking at. So in my case, the whole story started back at my grandparents' farm. This is the farm where I grew up, essentially. All my spare time was spent in this farm. There were meadows and forests, and it was really, really beautiful. You can see the forest in the background. This is what it looked like inside the forest. And I couldn't stop wondering what made this beauty, what made everything work so well together. Why is there such a harmony in nature? And why is it so destroyed when we humans interfere? So the answer to that is that everything is chemistry. So it's actually chemistry that communicates everything in this nature. We don't know the complexity of this chemistry yet. But it sparked my interest for chemistry, and while I was actually selecting that as a study later on. And when I look at young people who come to my laboratory and ask if they can have a position there, I could look at factual knowledge, and factual knowledge is still important, even though we have an internet that can allow you to be an expert in no time within any field. Expertise is very important because it's also the background for many of the other things. But what I mostly look on when I have to evaluate a person who is applying in my laboratory is whether you have intuition, whether you can imagine stuff. Are you good in associating the different knowledges you have into new ideas? Are you observant? Can you see that an experiment has an odd result that you really need to follow up on? Because that is where the new discovery lies. I also need you to be able to combine your ideas and make a platform, maybe be very creative, you know, utilizing computing as well as making new instruments and so on in order to solve your problems. Most importantly, I need you to have courage, particular courage to tell me that I'm wrong and that you are right, and that this is the way forward is your way forward. Because if you are able to actually argue and make an idea that holds water, you are on your track to become the next generation of researchers. But that road to becoming a researcher is never straight. Every time a PI defines a project, in particular in chemistry, it's doomed to fail. And that is also why you need to go to your PI and say, well, it just doesn't work what you're telling me, if it really doesn't work. And then you have to come up with an alternative idea that can lead to the result. You cannot plan because this failure is inevitable. So you cannot really plan, you have to go with the opportunities and see where the opportunities lies in your research. So you will fail and you will rise and you will fail and you will rise again with a vengeance, and eventually you come through. But time is very precious, so you have to do this. You have to have the intuition that tells you this is the right way, this is the right time to do what I need to do in order to get there. So get out of your comfort zone, learn new areas, and study for your entire life, bringing your expertise from one field into the other field, doing the research on the periphery between areas, because that's where the novelty lies. So I was very lucky to be offered a position at this prestigious laboratory. It looks a bit old, but actually we had all the infrastructure that is needed for top-notch research. We had 800 megahertz NMRs and all sorts of instrumentation, MSs and so on. So in this place I was allowed to do the research and we developed a new kind of material, the PEG-based polymers, which have the ability to function both in organic synthesis and organic chemistry, being lipophilic compatible as well as hydrophilic compatible, so it would work with enzyme and enzyme buffers at the same time. So we could do both the chemistry and the biology with these solid supports, with these polymers. So I started out at DTU as an engineer, so my engineering experience really followed me through my career and has been very important for developing a lot of technology. Then I was at Cambridge with Bob Shepard, a hero in peptide chemistry. I learned a lot of that and I went back and built a lot of instruments and so on. And then University of Copenhagen for a couple of years, and then 23 years at Carlsberg Laboratory, where I built the solid phase combinatorial chemistry center. And the idea here was that we took this support and we would do a lot of organic reactions and develop them into reactions that are just as efficient as when we built DNA and peptides. Then we would be able to synthesize any kind of molecule on solid support. And it was successful for all of these reactions that we have listed here. We have a large variety of publications that covers that. And it was also possible to do enzyme reactions. So we could actually do peptide cleavage reactions, glycosylation reactions, a lot of different enzyme catalyzed reactions that occur every day in our body, are important reactions, including immune cleavages and so on. So we had a whole range of different chemistries and biochemistries at hand. And this is the polymers. The most important one is the bigger polymer, but we also have polymers with no hydrogen source. That means that they can be used for strong carbanion and carbonium ion chemistries. And this is what they look like. These beads are made out of these polymers. These are small particles that is 0.5 millimeter in diameter, and you can see how uniform they are. So we can really use those in quantitative measurements of interactions between biomolecules and synthetic molecules on these resins. We were working within the big toolbox of organic chemistry back in 2000s, actually back in 90s, where we were working with this program. One of my PhD students, Christian Tornø, he was much younger than he looks here. He was involved in trying to make the Diels-Alder an orthogonal reaction to the peptide chemistry we were doing, so that we would be able to synthesize a peptide and stick on a side chain, for example, a fatty acid. As you might know, this is a very prosperous thing to do today because a lot of the hormones get a long lifetime in the body when they have these fatty acids on. So Christian tried to make this happen, and it didn't succeed. But instead he got another reaction, and this reaction was quantitative. It's the so-called Chuvak reaction, and that became the click reaction. It's completely regioselective reaction between an acide and an alkyne, and I'll show you what that means in a short while. Carolyn Bertozzi introduced the so-called spark reaction, which is a reaction between this cyclic alkyne and an acide. And then you have the TCO reaction, which is a tetrazine that reacts with a cyclooctene. These three are the best click reactions out there. There are other aspiring click reactions coming up these years. And in my opinion, we should develop as many as possible and get a toolbox upon toolbox upon toolbox of orthogonal reactions. Then we can build any molecular architecture that we want. So we also developed combinatorial method for split mix synthesis. That is, we want to make 1 million different compounds. We can do that by taking 1 million beads and then do 20 reactions, mixing up all the beads, putting them back there, do another 20 reactions, mixing the beads, and so on. Then it grows, the number of compound grows with 20 wells, so it goes with 20 times 20 times 20 times 20, and that becomes a whole lot of compounds within no time. Then we can treat those with an enzyme. If we have built an enzyme substrate that is fluorogenic, we can see fluorogenic beads as in this image, and we can pick those out and find out what the enzyme did, what was it active with. We have also developed a method where we immobilize microparticles inside the bead. So this is like a barcode. I don't know if you use barcodes here in the supermarkets, but if you do, it's simply a barcode that we can read every time we do a reaction, every time we do a screening of this particular bead, to learn what is the structure and what is the activity of the compound on that on the structure on that bead. So we have a whole platform technology that we have utilized over the years, and we are using that extensively for doing click reactions. We start out with the combinatorial chemistry. We generate a lot of different compounds, and you can see here some blue compounds on a green compound background. We select the blue ones, find out what they are. We resynthesize these compounds, and here we have injected it in a mouse. This is a compound that is highly active with one of the inflammation proteases, and you can see the the left foot pad on the back leg is inflamed, because here we see light from a fluorogenic substrate that is injected into this mouse. We can also grow cells on the surface of the bead to interact with the compounds that sits on the bead. And here you see a library of different compounds, and there are cells on all the beads over here. The green bead there is lighting up on a background of a lot of non-active beads. So the cells here are activated by the compound sitting on that bead. We isolate it and find out what the structure was and how it interacted with the receptor. And we can do that by the encoding technology. So that's a very powerful platform that has been used, as I said, for this quick reaction. A reaction between an acide and an alkyne to give us a product, triazole, that links two moieties together. This reaction is green because it can be carried out at 0.01 mole% of catalyst, very, very small amount. Each copper atom actually catalyzes formation of 10,000 molecules in water. So that is, of course, very nice, and also it's orthogonal, so it doesn't react with any other functionality in proteins, in carbohydrates, in DNA, etc. So what is it then? We have this triple bond, which is 2 carbon atoms with 3 bonds, so that's full of electrons because the bonds are electron density. And this electron density is looking out for some positive charge to combine with. And if we now add this positive charge in the form of a copper 1 ion, it will link to the electrons and it acidify the alkyne so that the copper sits on the terminal. Nothing much happened by that. But then a second one comes in, and that also attaches to the electrons. And now it has the power to actually peel off the electron density from the alkyne, leaving it very reactive as an electrophile. That is the LUMO of this compound. The empty orbital will react with electrons coming from the outside. And where can we get those from? Well, we want something that actually fits with this picture here. So the empty balloon should fit with filled balloons. And there's only one compound that really fit well with that, and that's the acide. So here is the reason for the selectivity and orthogonality of the click reaction. the reason that we got the Nobel Prize. It's right here. So they come together and we form first a metallocycle that loses one copper atom, then it loses a second copper atom, and then we have a product. And this product is not very exciting, but now if we took some very large molecules like these ones, which has real function, the first one could be an apoptosis promoter, which binds to the natural inhibitors in cancer cells of apoptosis or programmed cell death. And the second one could be one that locates the cancer cell by some cancer marker. And the third one could be one that enters into the cell. If we now take these and inject them, nothing will happen. But if we can combine them by attaching these click tags onto the surface, we can do that with a variety of different ways, from synthesis to amber stop codon suppression technology. We can get a molecule by steering overnight with copper 1, which is reacting only in one way, and giving one molecule, and that molecule we can then isolate and use, and now we can use it for finding a specific cell, for example a cancer cell, entering this cell via the small ninja that you see down here, and then affect the programmed cell death by this one inhibiting the inhibitor of cell death. So here we are in Stockholm. This was a very, very nice experience. 1 week of celebration of the Nobel Prize. The first day you are informed by the Nobel Prize Museum of the activities during the week, and then you deliver an item to the Nobel Prize Museum which is most important in your research. And this here is the first multiple column synthesizer in the world. Was very important for us. It's used everywhere in parallel synthesis machines, in combinatorial synthesis machines and so on today. These multiple columns. So what we discovered was that all these peptide reactions can easily be carried out in the open. We don't need to enclose it, and that made it very easy to make multiple column synthesis. So here is a signature, my signature on the top, right beside Emmanuelle Carpentier. I'm very proud of that because she's one of my heroes. This fellow is called a Japanese horseshoe crab. The Japanese horseshoe crab is a remarkable animal. It's 400 million years old, and it's like a pharmaceutical factory. We actually tap the beautiful blue blood, it's light blue color, and it has 2 macromolecules that can encage foreign organisms. So a bacteria that comes in is trapped. Then it sends in some toxins for the bacteria. These are like cannonballs that's shown up there in the corner. And we are very interested in how these work. So we would like to study those and to use click chemistry to do so. The natural peptide is a hairpin, so it's a bent peptide chain with 2 disulfide bond bridges. Now we would like to replace those with click reactions, because then we would avoid the normal metabolism of disulfide bonds in the organism. Is this possible? That's the question. Can we do that without damaging the activity of the compound? During this work, we found out by NMR, which is nuclear magnetic resonance, that these compounds actually form a dimer. It's a little cannonball that swims around, completely benign, with positive charge on the surface. As it hits the bacterial cell membrane, you see here the cell membrane, and as the cannonballs comes in and hit the cell membrane, they will divide into 2. So the dimer opens up and forms a pore, and through this pore the interior of the bacteria will come out and the cell will die. So that is how that works, and that's a very efficient antibacterial mechanism because it's entirely physical. So we wanted to try this on different organisms. Here is coli, staphylococcus, salmonella, typhimurium, and bacillus subtilis. These are organisms we would like to see the effect of this